At this stage in the course, you should understand how to select the proper delivery system to take a drug compound and make it into a drug product. Now I will discuss with you how to take a drug product and prepare it for clinical studies by gaining the permission of a regulatory agency to do so. This step of the drug development process can be quite expensive and can sometimes be an insurmountable hurdle for small companies and academic investigators. While regulators and researchers are trying to streamline the preclinical process by incorporating new technology and providing shorter paths for certain drugs, most developers still must invest several $million to pass through this clinical stage. While it may seem excessive that this much money is spent before a new drug can ever be taken by a person, all of this spending is necessary in order to provide as much assurance as possible that a new drug will not harm the first human subjects that receive it in clinical trials. In the United States, a new drug product must be given an investigational new drug designation by the FDA before clinical testing can commence. So what exactly is an investigational new drug, or IND? An IND is a drug product that has been developed and okayed for clinical trials but has not yet been approved for marketing in the U.S. by the FDA. It's essentially a legal designation that allows for transport of a new drug and testing of that drug in the clinical setting. The first step in this process is submission of an IND application. This is a single document containing information on a new drug product that will help ensure the FDA that the product is safe and ready for testing in the clinic. It is their job to ensure that a new drug will not expose volunteers to unnecessary health risks in Phase 1 clinical trials. To provide this assurance, information in the IND application must contain safety testing information, manufacturing information, and details of the proposed clinical study. If this information is clearly and adequately presented in the application, the FDA will open an IND application. At any point during clinical investigation, if the integrity of a study is compromised or the new drug proves to be a health risk, the FDA may put a clinical hold on the IND, halting any further testing in humans. The goal of this module is to provide you with a better understanding of how a drug product becomes an investigational new drug that is suitable for testing in healthy volunteers. Researchers often use living systems to determine if their compound will be able to move across cell membranes and impart its therapeutic effect. With many drugs, there is often a delicate balance between toxicity and therapy. Efforts are made to select a lead compound that has the lowest possible toxicity and the highest possible therapeutic potency. Much of this testing can be performed on the benchtop in cell cultures. However, these experiments are sometimes too simplistic to be able to predict effectiveness in a human. To gain a greater understanding of the efficacy and biological distribution of a lead compound, a more complex living system, typically a mouse or a rat, can be dosed with the compound and studied for a measurable therapeutic effect. Researchers have determined different ways to model certain human diseases such as cancer, infection, or inflammation in these rodents. While these models are not perfect predictors of human efficacy, they serve as another tool to select a lead candidate with the highest likelihood of success. Further, these models can also be used in studies to identify the optimal dose needed to achieve the targeted drug level. While demonstration of preclinical efficacy is not as important to the FDA as demonstration of preclinical safety, these efficacy studies can prove critical for drug developers when evaluating the likelihood of drug efficacy against a specific human disease. Once a lead compound has been selected and formulated, it must be evaluated in test animals to ensure that the product is safe for human use. While ethical objections often arise to animal testing, it is the best method scientists and government officials have developed for protecting human subjects from the unanticipated dangers of experimental drugs. These studies are required by the FDA and must be performed in a manner that is not only humane, but is also scientifically valuable. In order to ensure the validity and quality of the data from these safety studies, the FDA has put in place a strict set of regulations that developers must comply with. These regulations, called Good Laboratory Practices, or GLPs, set the organizational framework for testing a new drug substance and evaluating any toxicity that might occur during testing. Along with understanding any potential toxic effects, it is also important for the drug developer to know how long the drug remains in circulation, how the drug is metabolized, and which tissues it might accumulate in. If the lead candidate proves to be safe in these studies, the data can be submitted in an IND application as evidence that the compound is safe for human testing. In addition to this safety data, the drug developer must also demonstrate that they can produce a drug product that is manufactured in a controlled and reproducible manner.